Introduction to Galaxy. Let's see. I coordinate. Oh, yeah. Okay, good. So I coordinate outreach and training the community for the Galaxy project. Galaxy is a part of Anvil. There are many components, and we are one of them. So, and I have a computer science background, but I've been in biology about 20 years. But that lack of biology background will become apparent as we go. Okay. So I'm going to go forward. For me, I like questions as we go, but I can't really see things while I'm sharing my screen. So Stephen, Fred, Mike, Alex, one of you interrupt me, throw questions at me, or just people talk as we go, okay? Okay, so learning objectives. Today, we're going to learn how to launch Galaxy in Anvil. And Galaxy is well, yeah, we'll get to that. And then we're going to import data from Terra, which is the orchestrator for Anvil, as we learned yesterday. And then we're going to learn the basics of the Galaxy interface. So that's our goals for today, are those. So let's start out with what is Galaxy. This is the project's definition. Galaxy is an open web-based platform for accessible, reproducible, and that reproducible is very important, and transparent biomedical analysis. This statement reflects our values, and it's all true, but it's not how a lot of people actually think of the Galaxy platform. So for that, I'm going to use Keith Bradnam's definition. Keith says, Galaxy is a web-based platform that provides a simplified interface to many popular bioinformatics tools. And this is how a lot of people view Galaxy. We protect researchers from the command line. We protect you from having to learn Linux systems administration. You don't have to install libz to get some other package to work. You don't have to learn all that. You can just use Galaxy. So both of those definitions are true, the one from the project, the one from Keith, but it's only half the picture. And the other half of Galaxy is the community. This quote from James Taylor, founder and Galaxy PI at our 2019 community conference: the most important thing about Galaxy is this community. And I really would like to stress that our community is absolutely vital to our success. Without our community, we would not be the presence that we are. We would not be viable. So we do have an enormous and awesome contributor community. And here are some examples, and we could go on and on. I like the middle one in particular from Black Duck Open. This is about the source code, not about the rest of the ecosystem. Over the past 12 months, 156 developers contributed new code to Galaxy. This is one of the largest open source teams in the world. Okay. So some of us are funded to work on Galaxy. Some of us use Galaxy and then contribute back to it. And some of us just do this as a hobby. So again, the community, the importance of the community cannot be overstated. Okay. So Galaxy is a platform. How is it available? And it's available in lots of different ways. First of all, it's available through the web. And that's the main way people will access it, is through your web browser. That's what we're going to do today. But where on the web is it available? Well, it's available in lots of different ways. There are public servers. So people have installed Galaxy. It's open source software. Anyone can do this. They have installed Galaxy and installed tools and then put it up on the web and made it available to anyone who wants to use it. And it turns out there are over 130 websites that are based on Galaxy right now. And some of them are pretty big, like the usegalaxy.star ones, usegalaxy.org. They're pretty big. They have a lot of compute resources behind them, a lot of tools, a lot of reference genomes. They're also shared resources, which means when you use them, you're sharing them with the world. So on any given day, they might be responsive, they might not. And they also impose quotas. So because again, they're shared resources. So keep that in mind. Second option is you can run Galaxy on your laptop. And this just takes a few minutes. There are a number of containers, which means Docker in our world mostly, that have a ready-to-run Galaxy already installed in them. All you have to do is download that container and then launch it. And then you have a full-fledged Galaxy instance. It's also available on the cloud, and that's what we're going to use today. It's available on a number of national research infrastructures, including Anvil and Jetstream in the US. Around the world, it's available in many countries. So Nectar, Climb, and GenAP, for instance. Nectar is in Australia, Climb is in the UK, and GenAP is in Canada, where you can launch a ready-to-use Galaxy instance on a cloud infrastructure. Okay. Last option here is your organization's own infrastructure. So if none of these options work for you, you can launch it on, well, you can bring it up on your, it's possible to bring it up on your own infrastructure. So if you have a high performance computing cluster, the people who run that can bring it up and make it available to you. We think this is the most common use case. So it's used in a lot of hospitals, things like that. It's behind the firewall, okay, for that. So lots of options. What makes Galaxy on Anvil special, particular? As it was talked about yesterday, Anvil takes Galaxy, or the compute, to where the data is. So the data is on the cloud. And Anvil runs compute on the cloud, and the data is now enormous. So this actually matters, because if you're doing large analyses, moving around terabytes becomes an issue, becomes expensive too. So that's one reason. Another is that Anvil runs Galaxy in a FSMA moderate setting, which means you can now use public cloud infrastructures because Anvil runs on Google with restricted data. So if we go back to this slide, these first three options, mostly they're semi-secure, but they're not certified in any way. They're not HIPAA, they're not CLIA, they're not FSMA, they're nothing. Okay, we do our best, but it's not guaranteed. Here it is FSMA moderate. It has been certified for that, and so it allows you to use restricted data on scalable public cloud infrastructures, and that is a huge plus for Galaxy on Anvil. So secure access and public cloud infrastructures. Okay. Another thing it offers is integration with Gen 3 and DocStore and Terra. Okay, and we saw those yesterday. I'm getting feedback from someone, but I don't know who. So let's see. So we also get integration with the whole Anvil, ecosystem. And these features are also true for our sister platforms in Anvil, which is Bioconductor and Jupyter Notebooks. Okay, so all these things are true for those as well. Okay, let's spend a little bit on the ecosystem. Yep.
Excuse me, Dave. I had a question. Yes. This is Helena.
Yes, thank you, Helena.
Okay, how are you?
I'm good. I'm really glad you're on the call because you asked great questions yesterday. So.
Thanks. What is FSMA, F-I-S-M-A?
Oh, Mike, can I throw that at you? Mike's.
Sure, happy to. So, FSMA is sort of a level of certification that you have to kind of get accredited for. That involves a lot of sort of encryption technologies, authentication technologies, logging technologies, intrusion detection systems. Basically, what it is sort of a stamp of approval to say that this platform is certified to host protected data sets. As you might imagine, there's a lot of concern from us, especially in outside the world, that human genome data is very, very sensitive and is treated very, very carefully. The analogy I often say is, if someone steals your credit card number, it's a big pain and it's concerning, but we can issue you a new credit card number. If someone steals genome data, I can't give you a new genome. So we've gone through incredible levels of scrutiny and certification to make it, as hardened as absolutely as possible to prevent any sort of unauthorized access to the data, any sort of breaches. In sort of government parlance, this is called FSMA moderate certification. it's the level of certification that we've reached for that.
Thank you, Mike. Thanks for the question, Helena. Keep them coming. Okay. Okay, so let's talk about the ecosystem. There is a huge ecosystem around Galaxy. Here are some support options. We have an online forum that you can post things in. That's based on discourse. Anvil is now using discourse as well, so it's a great way to ask questions and get answers. If you prefer chat, we have a whole bunch of places you can go for chat. And then finally, Anvil Support Hub. This is all sorts of things about Anvil. So if you have specific questions about Anvil. Okay. Training. There is a lot of training resources out there. So that's the URL for Anvil specific training. Galaxy has a lot of training events, and more importantly, I think we have the Galaxy Training Network, which is a huge library of training materials that you can use on your own or in a classroom setting to learn various parts of Galaxy and various parts of bioinformatics. So really, that is a spectacular resource. And that's run by the community, not by me, by the US project. It's actually run by the Galaxy community. Okay, so here's an example of the Galaxy Training Network. If you go there, you land here, and it says, hey, there's stuff for scientists. And you go in there, Introduction to Galaxy Analysis, takes you here. There's about, I don't know, 10 intro tutorials. Peeks the Gene is a good one. You go there, and then within Peeks the Genes, there's all sorts of hands-on exercises. And so these tutorials are a mixture of information about like peak calling, okay, as well as mechanics, how to. So here's what you want to do to solve this particular question. So again, a spectacular resource. Okay. Some more resources. The Galaxy Community Hub has events, news, you name it, it's there. The Toolshed is a place where people, they wrap tools for Galaxy, which means you can import them into Galaxy. We might see this later, but I don't think we'll have time. One cool thing about Anvil is you can install your own tools on your own Galaxy through the Toolshed. So we'll see if we get to that. If we don't, it's in the tutorial. And finally, Anvil, of which Galaxy is a part. Same sort of things, that are here, except for the whole Anvil community. Okay, so let's get to the hands-on part. We're now going to start Terra in Galaxy. So what's here in the slides is a bunch of screenshots of what we're going to do. I'm not going to spend a whole lot of time there, okay? What I'm going to do is just walk through in the other tab, much like was done yesterday. So we have the slides as a backup, but we're just going to do it, okay? Boom. So you want to go to anvilproject.org. I'm going to zoom in a little. Okay, that'll work. Is that okay? No complaints. This is good. Okay, we're going to launch Terra. So we click on Launch Terra. Okay. And then we want to sign in with Google. I'm going to do that. And it's going to come up and say, hey, Dave, who are you? And I want to log in with my account that I have used for Terra. So when I created my Terra account, that's the one I want to use. So I click on that. And it says, Dave, you're golden. It lands me here on the Workspaces tab. This is good. What we want to do once you're there is go to public. So something else that's common today with yesterday is like yesterday, I'm not going to be able to come back and guide you step by step if things get lost. So if you fall behind or if something goes wrong, what I really recommend you do is stop and just listen, because the screenshots are all there. The tutorial explains things. You can come back later. Whereas if you're spending your time trying to catch up, I think you're going to miss a huge amount. So I apologize that I can't provide that level of support. But if you actually do fall behind, think about just giving up and, listening to the presentation. Okay. So we landed in Anvil and Terra, and we got here and we clicked on public. This is good. And now we're going to search for the workspace we want to clone. Again, just like yesterday. And I searched for message. And I came up with only one in public, Galaxy demo message in a genome assembly created by Fred Tan a couple weeks ago. So we got that. That's good. I'm going to go over here. I'll zoom in some more. I'm going to go over here and click on this vertical ellipsis right there. Boom. Clicked on that. And what I want to do is clone it because I want to use this workspace to run today's analysis. So I'm going to clone that. And it comes up and it says, Dave, what do you want here? And it's like, okay, Galaxy demo message in a genome assembly copy. Okay, well, that's true, but it's not that helpful. So I'm going to say, this is Clements, and I'm going to say VADSTI dash, okay? Now, if you are actually using Anvil in anger, so you're doing a lot of stuff in Anvil, you need to pay attention to what this workspace name is, because it has to be unique within your billing project. Now, for all of you, Anvil's brand new, so you're not going to have collision problems, but you can't have 3 people in the same billing project create exactly the same workspace name. You all have to have unique. So putting your name on the front is not a bad idea in general. Yeah. And I'm going to select my billing project. Come on. And I've got a bunch because I work for Anvil. And this is the one I want to use. It should look a lot like the one you have right there. Boom. I'm going to click on that. So you should only have one billing project because you're new. I'm going to take the description exactly as Fred created it. That's fine. And since today we're using entirely public data, I'm not going to worry about authorization demands. See yesterday's presentation for more about what that means. And then we're going to clone the workspace. Okay, so unique name, set my billing, clone. Okay, so here we are. We got a couple things. Let's play around with it. I'm not spending any money now. That's good. Let's click on data. And we saw some data tables yesterday. This is the one that's going to matter for us. Because we have one data table, it's called sample, has 5 rows in it. So this is just a spreadsheet, basically, okay? But these are special. And what makes them special is that this column contains URLs that point off into Google Cloud, okay? And Anvil recognizes, hey, this is a cloud resource. And it means it treats it special. If I click on it, it tells me here's what it is and here's where it is in the cloud. And if I have GSUtil installed, I can download it. Okay, great. But we're going to use these 5 once we launch Galaxy. Okay, so we'll come back to that. We don't have any reference data or other data or workspace data or files. But now we're going to go to notebooks. And notebooks is where we launch Galaxy. It's also where we launch IPython. And we're just going to click on this. Okay, create a cloud environment for Galaxy. We do that. It comes up and it says, okay, Dave, this is going to cost you 53 cents per hour. And if I pause it, okay, so it's going to cost me 57 cents an hour, that plus that. Okay. And I got a configuration here. I'm okay with the minimum. And I encourage you to just stick with that because it'll come up faster. One thing to note is you have to have at least 300 gigabytes or else it won't launch. So I'm just going to go with the default of 500. I'm going to click next, and then I'm going to click create. Now what's happening behind the scenes is Anvil has started to launch Galaxy, and it's going to go through 3 steps here. Once it's done, we're going to have our own instance. So each of us is going to have our own instance of Galaxy that we have complete control over. Okay. And wrong one. There we go, there. Okay, now we're back here. What's going to happen next? It's going to take a few minutes to launch. While that happens, Mike is going to introduce, genome sequencing, as well as the question we're going to ask today, and then we're going to come back and answer our question in Galaxy. So I'm going to stop sharing. And Mike, I'm going to pass it to you.
Okay, thanks, Dave. Thanks for that great setup. So what we're going to do now is so we just saw how to launch Galaxy. In reality, it was just like, I don't know, a handful of clicks to get it running. It does take a few minutes to get that set up. So what I'll do is sort of give you this sort of classroom lecture about some of the genome analysis that can take place. My understanding is, we have a very diverse class. Some people are very expert, some people are quite novice when it comes to genomics. So hopefully there'll be something for everyone in this presentation to kind of walk through some of the concepts. So let me go ahead and share my screen. Oh, I have to set some system preferences. Sorry about this. This will just take a minute, hopefully. Allow this. Unfortunately, I'm gonna actually have to sign off. I didn't realize this. The way that Mac's permissions are set up, for me to give permission to WebEx to see the screen, I have to quit, but I will be back in just a jiffy. Dave, maybe you could field a few questions while that's going on. Sorry about that.
Yes, no worries. So the slides Mike is going to show are in the agenda that you should have received in an email from Stephen Mosher, as is the tutorial and the slides that I'll use this later today. So any questions? Manuel? No. Okay. Yeah. So I am gonna, until Mike comes back, I am gonna emphasize what makes Galaxy on Anvil unique. And really the protected data is a big one because it is a secure environment. And again, other options, they don't have that certification. Second thing, until Mike shares, is the ability to scale. So when we launched Galaxy, we used the defaults, but we could actually make that server huge. Oh, I've been talking the whole time and no one could hear me. Crap.
No, we can hear you.
Only the last sentence. Only the last sentence. It can scale and you can make it huge. We heard until that point.
Yeah.
Okay, good. Okay, Mike, you ready?
Yeah, let me go ahead and share.
Learning WebEx.
Okay, can everyone see my screen?
Yes.
Yeah. Great. Okay. Yeah, so today I'm going to be talking about genome sequencing. So that's the process where we can read off molecules of DNA. And then I'm going to be talking about sort of a common sort of analysis that takes place after that, right? So with all sequencing technologies, we can't actually read entire chromosomes. There's a process where we take these individual reads and kind of stitch them together. That's this problem. That's this problem called de novo genome assembly. So I'll be talking a little bit about the sequencing and then how we do assembly to actually reconstruct a genome for further analysis. And then in the exercise, we'll do those steps. We'll also do a bit of gene finding to kind of look for novel genes in this very special strain that we've put together for you. Okay, so what I'm going to do, because I think a lot of these concepts are...
Hi, Mike. Let me interrupt before you continue. A very brief self-intro.
Oh, yeah, I'm sorry. Great. Yeah, so my name is Michael Schatz. I'm an associate professor at Johns Hopkins in computer science and biology. I'm also one of the co-leaders of the Anvil project. Kind of like Dave, I've been in genomics coming up many years, almost 2 decades now, with sort of a split background, like I said, between biology and computer science. So because I think some of these topics are going to be new to some of you, I'm going to start with what I hope is a pretty gentle introduction to some of the the fundamental concepts. I like to do this by this little cute analogy. Please bear with me. I think it'll make sense when it all comes together. Then we're going to talk about some of the more specific practical issues when it talks about if you really want to sequence a genome, here are some of the things that you really got to pay attention to. And then in the last 5 minutes or so, I'll introduce this exercise where we'll be doing a live analysis inside of Galaxy, where we're going to be assembling a genome, doing whole genome alignment, looking for novel genes on a novel structural variant. Okay, so I think this class probably doesn't need this motivation, but there's tremendous interest to sequence genomes because genomes carry with it so much rich information about who we are, right? Almost every trait has some association with your genome, like things like how tall you are highly heritable. If I know how tall your parents are, I have a pretty good idea about how tall you will be. Other characteristics, sort of physical characteristics, susceptibility to disease, even sort of cognitive considerations, health considerations. There's just enormous interest for many traits and diseases to be able to sequence genomes so we can sort of take a look at a genetic level, what sort of risk you have, what sort of predisposition you have. I'll be the first to admit, though, your genome is not your destiny, in that your environment, your experiences, your exposures do play a critical role. There's often this very tight relationship between sort of your molecular characteristics in your genome and then your whole life. So it is not your destiny. You're not your destiny, although it does. Your genome for sure does play a major consideration. Hey, Emmanuel Akala, I'm getting a little bit of background noise from you. If you can mute yourself, great, thank you. Okay, so there's tremendous interest to sequence genomes. There's been
surgery center queue.
Yeah, Emmanuel. Yeah, you're still getting some feedback from me. So there's enormous interest to sequence genomes, and then over the years there have been a few major technologies that have been developed and put forth to do that sequencing. So back around the heyday of the Human Genome Project, this was so-called Sanger sequencing, named after Nobel laureate Fred Sanger, who invented the protocol for doing the sequencing. Those technologies today are more or less obsolete. They're very slow and expensive. They're very limited. You might use them for critical validation of clinically relevant variants, although even that application these days is not widely used. Today, by far the most widely used platform for DNA sequencing is the Illumina platform. That's the name of the company. The name of the technology is called sequencing by synthesis, and I'll give some more details at that in a little bit later. But kind of the key characteristics is that it's quite inexpensive. For a few hundred or maybe $1,000, we can sequence a whole genome. It's at very, very high throughput. More than 1 million genomes have been sequenced with Illumina sequencing. I think it's like we're approaching 1 million genomes a year with Illumina sequencing. It's that sort of capable. It's very low error rate, so very high quality. The main sort of trade-off for it, though, is the reads are quite short. A typical human chromosome is a few 100 million nucleotides, but it can only read off a few hundred nucleotides at once. So we need to collect many, many, many sequencing reads in order to get across an entire chromosome. The newest platforms for sequencing, so-called third-generation sequencing, use what's called single molecule sequencing, where individual molecules of DNA are isolated using different technologies and read off. These are really kind of emerging. They're starting to become very popular. They bring with them some very special capabilities. At the top of the list is that they provide much longer read lengths. Whereas Illumina sequencing is limited in practice to a few hundred nucleotides, today it's very, very common to sequence molecules that are tens of thousands, hundreds of thousands, even millions of nucleotides in length. And again, if we want to get across a whole human chromosome, say 150 million nucleotides long, having reads that are like 1 million bases long makes this a much more efficient operation. Now, historically, these instruments have been sort of slow and expensive. Shown here is the PacBio RS2, which even today, this is the sequel to, and even today on this instrument, you can only sequence a few hundred genomes a year per instrument, whereas a single high throughput Illumina sequencer can sequence thousands and thousands of genomes per year. So it is a bit slower in that regard, although it's getting faster. And then also there are some newer technologies that are emerging that have increased throughput. Actually, so this is another single molecule sequencer. This is the Oxford Nanopore MinION. This costs about $1,000 and can sequence a whole human genome in about 2 days. So if you need to sequence many genomes, well, you can just get many Oxford Nanopore devices and sequence them very efficiently. The challenge for these new technologies is it does have a substantial error rate. The raw read error rate is a few %, although as you'll hear about next week from Adam Philippi, using clever software algorithms, we can take that few % error and basically drive it to almost no error whatsoever. Some of the new genomes that we've been assembling with these technologies, we estimate the error rate to be better than 1 in 10 million nucleotides. In fact, it exceeds our ability to detect error at this point. We basically can get perfect genome assemblies with these new technologies. However, with these technologies, all of them, they carry, like I said earlier, they carry the big limitation in that you can't actually sequence the whole chromosome at once. We're going to be sequencing different sort of snippets of it, and then we need to then sort of piece them together to be able to reconstruct the whole genome, kind of telomere to telomere, end to end, out of the raw sequencing data. So we have this problem. You know, we absolutely want to sequence to study health, genetic diversity, all these sort of medical conditions and so forth, but we can't actually sequence whole genomes. So we need some software. We need some algorithms that can compute this process. Now, the name for this is de novo genome assembly. It's very, and that's going to be our main topic for today. But to kind of introduce some of the ideas, I'm going to sort of present this little analogy that will hopefully be very digestible to kind of lay out some of the concepts. So I call this the shredded book reconstruction problem. So it goes like this. So imagine you're Charles Dickens, a couple hundred years ago, and you just finished writing A Tale of Two Cities, right? So this is this very famous book that begins, it was the best of times, it was the worst of times, it was the age of wisdom, it was the age of foolishness, no. So Dickens just finished writing his masterpiece. He wants to make lots of copies of it so he can go, sell it and distribute it to the world. Here's the one little tricky part. Rather than printing it up on a piece of paper, we're going to get it printed up on this very, very long spool of paper, right? So hopefully this is my very transparent metaphor for, a very long molecule of DNA where there's sort of characters written on it. Okay, so Dickens, he writes it. It's on this spool of paper. He goes to the copy shop. He gets many copies of the book printed up. But as he's walking out of the copy shop, he trips, and then it falls into the paper shredder. And then those very long spools of paper get cut up into these very, very short fragments. So instead of having, the whole book written out, now it's going to be chopped up into like 5-word fragments here. If you've ever opened up, it's kind of silly, but if you've ever opened up like a fortune cookie and get that like little piece of paper, that's where we're going to have to work from after it goes through the paper shredder. So it goes through, and then all of these little fragments of paper are in this shredder bag. They're all mixed together. A Tale of Two Cities is about 140,000 words long. You can go look it up. So if we have 5 words per fragment, 5 copies, we have like 140,000 little fortune cookie sized pieces of paper that are all jumbled together. We've lost track of, where's page 1, chapter 1, book 1, all of that's mixed together. We just get this, this big bag of pieces of paper. So, just like Dickens, oh no, we want to put it back together. We need some strategy to do so. Now they're all mixed together. We've lost track of where they come from. And then one really critical complexity is that some of the fragments that we're going to see in a second are going to be identical. Now, if you're cutting up these pieces of paper and you happen to cut at exactly the same place, yes, the fragments will be identical. But notice here, there are fragments that come from different positions in the book that have exactly the same words on them. So of times it was, that occurs twice. It was the age of, that occurs twice, even in the same sentence fragment. And that's going to be crucially important in just a second. So what I have here now is it's all jumbled up, and we need some sort of strategy to put it together. One useful thing to do is just write it, take all those fragments, and then just put them in alphabetical order. There's a lot of practical reasons why that's good. It makes it easy to search. It just makes it so you can sort of thumb through them quickly. Although in itself doesn't actually lead to a reconstruction. Now we just have this big table of 140,000 fragments that we want to go through here. So now what we want to do is we want to take this big table and then try to organize it and then sort of piece it together. If you ever put together a jigsaw puzzle with like little tiles, it's very analogous to this process here. So the strategy that a lot of software uses, in fact, all of the early genomes were kind of put together in this way, is a so-called greedy reconstruction. Let me explain what that is by an example. So what we're going to do here is we're going to reach into this big table of fragments, and we're going to pick a starting fragment. Now, this can be done sort of randomly. This can be done sort of arbitrarily. We'll just pick the first fragment. You may want to do some analysis to try to pick out fragments that occur. Maybe some occur a lot, some occur infrequently. But by some rule, we're going to identify some fragment inside of this much larger table of 140,000 fragments. So here I've just picked this one. It was the best of. Now, in some regards, the whole business of genomes assembly, or this book reconstruction problem, is to sort of, from a given fragment, ask the question, what fragment comes next? It's, again, we're putting together this jigsaw puzzle. We have a piece of the jigsaw puzzle. Which other piece sort of locks onto it? Now, the jigsaw puzzle, the pieces lock together based on their physical shape. But here, the jigsaw puzzles are going to link together based on the actual words that are written on them. In the case of DNA sequencing, it wouldn't be words, it would be the actual nucleotides where we want the nucleotide sequences to match up here. Now, in principle, we can find these matches in many different ways, right? All we know is this fragment here reads, it was the best of. You could imagine finding some other fragment that overlaps by of, or best of, or the best of. In general, though, we want to find fragments that overlap by as much as possible. So I can scan through my list here and I can find, aha, here's this other fragment that also has 5 words on it. And then the end of the first fragment matches the beginning of the next. That's what an overlap is. They overlap by 4 words. So taking these 2 fragments, each 5 words long, we've started to reconstruct. Now we've actually spelled out 6 words. It was the best of times. And then, like a good computer scientist, we just repeat. We've sort of figured out how those first 2 pieces lock together. Let's repeat. Let's try to find another fragment that locks together in the sense that the words match up to build out those overlaps. So now I have 3 fragments that I've been able to spell out the first 7 words to reconstruct this book. We can just repeat, 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 no problem. The way I think of this is if you think about like little Lego bricks that each have, I don't know, I have a 5-year-old, so Legos are my world. If you think about Lego bricks that have like those 4 little dots, well, we get those 4 dots, connects to the next 4 dots, connects to the next 4 dots. I can build a Lego bridge that will go around the world or make it to the moon, assuming I have enough Lego pieces that all lock together. But some interesting things start to happen. So here's an example where there are 2 fragments that overlap equally well, but those fragments have exactly the same words on them. They both say of times it was, no problem. We can just lock them together. They all get connected equally well. We can just connect them, and it's unambiguous how everything should be put together. However, as we go to the next fragments, we get a conflict. So here are 2 fragments that overlap equally well. They both start times it was the, but those fragments conflict. They do not share the same words at all. They start to really go to different parts of the book. So it's ambiguous. From this process, this greedy reconstruction where we just iteratively try to add on, add on, add on, what really happened, if you remember in yellow, those were fragments that were duplicated. They were repetitive in the original bag of fragments. So we started in unique, we walked into this repeat, and then as we walk out of this repeat, we're not sure where we should go. It could go, it was the best of times, it was the worst, or it could go, it was the best of times, it was the age. We just don't know. We can't tell using this greedy process. So whereas early genome assemblers, FRAP, Tiger assembler, early assemblers, even using the Human Genome Project, they can partially reconstruct the genome, but then they get stuck. That's where we need sort of a smarter algorithm, a smarter technique to get through this. Over the years, there's been a few approaches that have been proposed. A very, very widely used and popular one today has a funny name. It's called the Bruin graph assembly, the Bruin graph construction, named after a mathematician in the 1940s that developed some of these fundamental ideas in a pure math sort of context, although they get applied extensively in genomics. So it started as a pure math concept. It started in graph theory, although let me sort of walk you through it with a concrete example. So the way that this graph is going to get constructed, this sort of representation of how all the sequences relate to each other, is we're going to take those 5-word fragments, like it was the best of, and we're going to do something counterintuitive. From those 5-word fragments, we're going to derive even shorter fragments. We're going to derive, in this example, 4-word fragments. So from that first fragment, it was the best of, we're going to derive, it was the best, and then the second 4-word fragment was the best of. It's a very simple operation. In, I don't know, 5 lines of Python or 5 lines of R or 5 lines of C or Java, you can very, very easily take a DNA sequencing read that's like 100 nucleotides long and then sort of chop it up into the individual subwords. Those are sometimes called k-mers. And then the trick of a de Bruin graph assembler is we just keep track of which of these 4-word fragments, which of these small words is connected to which other 4 words within some read. So we have in the first read, we have it was the best of is connected to was the best of. And then in the second read, we have was the best of is connected to the best of times. And then we just draw this little picture that connects all those different short fragments together. So it seems very simple, and indeed, computationally, it is very simple. But in a second, hopefully, we'll see why this is a good idea in order to help us reconstruct the whole genome or the whole book. Let me pause here and just sort of check in and see if there's any questions. Okay, so we can go, it'd be very tedious to do by hand, but we can very easily go through our list of 140,000 fragments, break it up into these little subfragments, keep track of who's connected to who, and try to model this as this large data structure. So let me show you a proof by picture that this is a good idea. So this is me sort of making a little animation, simulating this process that Charles Dickens could do by hand, although admittedly it would be very tedious. So once you do this, notice here, all of these nodes in this graph all have 4 words on them, and then they get connected to other nodes that have 4 words on them. Most of the time it's unambiguous, where you go from one to the next, but occasionally you get those branch points. And if you look at our original sentence where we know the answer, those branch points occur around repeats. So this is what makes this a complicated process. So right here, this would be the De Bruin graph of that first sentence of A Tale of Two Cities. It's a very simple idea. Again, in 10 lines of Python, 10 lines of R, you could develop this graph, although scaling it up to an entire human genome, you're probably going to want to use a more concise representation to do this efficiently. So once you have this initial graph, everything is 4 words long, so it's hard to see the original book, but some parts of it are very easy, and some parts of it are very understandable. So you want to sort of compact or compress down this graph, which we can do here as much as possible. So we're going to take this initial graph where everything is of uniform length, and then we compact it down into this much more concise representation where we can sort of piece together all of the parts of the book or all the parts of the genome that can be unambiguously represented. So we have, for example, up here we have it was the best of times it was unambiguous how that gets represented. So let's go ahead and do this. This is exactly what happens inside of a genome assembler, where we're going to get millions or billions of short reads. We build up this graph and then we compress it down, compact it down into the parts that are unambiguous. That will be your initial assembly result of the parts of the genome that can be unambiguously reconstructed. Hopefully that's successful, although in practice rarely is. You're not going to get entire chromosomes end to end through this process. You're going to get these loops, you're going to get these branch points that remain because of repeats in the genome. Now here, if you're looking at this graph, you might be tempted to say, oh, this is easy. Obviously it starts in the top left, and then obviously it cycles through. We want to visit every node and visit every edge. And so it's obvious how this should be connected. Now in English, it would be extremely awkward to write, it was the best of times, it was the worst of times, it was the worst of times, it was the worst of times. It was the age of wisdom, age of wisdom, age of wisdom, age of wisdom. It was the age of fools. It'd be extremely awkward to write that in English, although in genomes, those tandem repeats do occur all the time. So we have to be really, really careful about how we process this graph any further. You want to be very conservative. You only want to make modifications to the graph that you can be certain are not going to introduce any errors. And then furthermore, if this is just the first sentence, if you look at the entire book, for whatever reason, Charles Dickens really likes this sort of sentence structure. So these graphs very quickly start to become very, very complicated as you're looking over the whole book or the whole genome. In fact, if you look at real genomes, so this is a simulated experiment from a few years ago, where this graph is just looking at the microbe E. coli using 50 base pair subwords of the genome. You start to get this really complicated graph of how the different segments are related to each other. And again, it's all because anytime you see those branch points, that's where there's repeats in the genome. So, these graphs in some sense represent the complete repeat composition that is present there. And that's sort of the rest of genome assembly, is trying to figure out how can you move beyond and beyond through these different repeat sequences that are present. So one sort of quick aside is we can build these graphs. We can try to compact them down as much as possible. The examples I've shown you have been pretty simple, where maybe it's just one cycle or a few branches. But these graphs become very, very complicated very, very quickly. Right? So this little clover leaf up here at the top, where you have some unique segments A, B, C, D, with some repeat in the middle. There's basically two different genomes that you could spell out here. One that goes A to the repeat to B, then to the repeat to C, then to D. Or you can go from A to the repeat to C, then to B to D. There's only really two solutions. But if you imagine you had that clover leaf followed by another clover leaf attached to another clover leaf, you very quickly get into this combinatorial explosion where you have a two-way option followed by 2 by 2 by 2 by 2 by 2 by 2. It can branch very, very quickly into many, 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 many possible options. A few years ago, we did an analysis. You can actually work out numerically how many options there are. And we did an analysis of all the microbial chromosomes that were available at the time as to how many possible reconstructions they are using short-read sequencing. Notice here, so the x-axis is the length of the chromosome, the y-axis is the number of reconstructions, and then this is measured in powers of 2. So for a chromosome that's a few million bases long, there's like 2 to the 1,000th possible reconstructions. That's an incredibly large number. It's very unfamiliar. So what is 2 to the 1,000th? This is like 10 to the 300th possible solutions. There's only 10 to the 80th atoms in the universe. So from a small genome assembly problem, the sort of information complexity actually exceeds the number of atoms in the universe. It's just like incomprehensible how complicated genomes are, even for a microbial genome, never mind the human genome that would be many times bigger. So, from a genome assembly, especially from short reads, the process is very hierarchical. We're going to chop them up into even shorter reads, lay out how they're all related to each other, try to compact it down. We're going to try to do this over the segments that are unambiguous, although inevitably there's going to be repeats that are going to make it ambiguous. And we can look for, some possible solution, although in practice we need additional data to actually write down our entire chromosome end to end. You just can't do it if all you have is short read sequencing data. What I like, kind of playfully, normally when we think about a hard problem, we say, oh, this is astronomically hard. But now I would argue we should be thinking about this as a genomically hard problem, which is orders of magnitude more difficult, just because of the information content that is present there. So that's sort of the conceptual framework. Again, let me pause here and see if there's any questions before moving on to some of the practical considerations.
No questions in chat yet.
Okay, well, feel free to jump in at any time. I'm going way slow, so let me try to jump ahead now to some of the practical considerations. Okay, so let me, this is just for your notes where you can see how a small genome could be reconstructed, where we go from forward fragments, derive them into k-mers, build the graph, and that will spell out Gattaca. So that'll just be for your notes, so you can sort of review how that process works in the case of nucleotide sequences. Okay, so let's move on to some of the practical considerations, like how are genomes actually assembled? How are they actually pieced together? What are some of the trade-offs that you might see there? Okay, so first thing to note is that genome assembly comes up in lots and lots of applications. Here are kind of logos from the Genome 10K project. There's the Insect 5K project. There's the Arabidopsis 1001 Genomes project. And it kind of goes on and on and on and on. Now that genome sequencing has gotten relatively inexpensive, there's great interest to sequence basically all living organisms. At some level, they're interesting. They basically all need genome assembly in order to establish a reference genome in the first place. So there are literally thousands, tens of thousands, hundreds of thousands of projects underway to sequence all these different diverse species. Another great example of genome assembly is so-called metagenomics, where you're not going to be sequencing, isolated species. You're going to be going to a complicated environment. This could be, all the microbes that live on you or maybe inside of you. We can't isolate them one by one, but we're just going to be sequencing and assembling all of them all at once. This is the problem of metagenomics assembly. It also uses the Bruin graph assemblers. It also does a lot of the same consideration for repeats, although there are specialized software packages that you should use in the case of metagenomics. Here I have logos from the Human Microbiome Project, over thousands of people. They're looking at many different body sites there to characterize the microbes that are there. There's been some exciting results there. In a model system, they've done experiments where you take skinny mice and obese mice, and you swap their stomach contents, and skinny mice become obese and obese mice become skinny, clearly the microbes living inside of mammals, including us, play an important role in metabolic processing. The latest research there, it turns out microbes are that live inside of our gut are basically pumping out neurotransmitters like serotonin and other related chemicals. It's somewhat mysterious, to what extent those neurotransmitters are influencing our brain chemistry, but I'm willing to bet they're playing a huge role. It's just not really well understood today. Another example here is shown from the Sargasso Sea project, where you just basically, in the same way that we can point telescopes into the heavens to look and just sort of catalog all the stars, all the galaxies that are out there, we can point our DNA sequencers into the environment and just characterize all the microbes that are seen there. Through these projects, we have greatly expanded the number of known species and greatly expanded the number of known genes, and by consequence, the number of proteins in the world, just by doing these great surveillance projects. And then finally, these assembly concepts to Bruin graphs and overlap graphs and sequence graphs come up in many, many other contexts. In addition to sequencing genomes, maybe you're interested in sequencing transcriptomes, looking at RNA. Maybe you're looking at whole collections of genomes all at once, population genetics, graph genomes. A lot of these same technologies get applied in these different applications as well. So genomes assembly has been around basically as long as we've been sequencing genomes, back to the 70s, but it remains a hard and interesting problem to this day for a number of different reasons. Biologically, humans are kind of boring. We're only diploid, but if you look around the living tree of life, there's a lot of species that have much more complicated genomes, especially plant genomes tend to be much more complicated. So the wheat that we eat in our bread, for example, instead of having 2 copies of every chromosome being diploid, has 6 copies of every chromosome. It's hexaploid, and it has a much larger genome as in addition to that. Other complications is all DNA sequencing platforms have error associated with it. We need to overcome that. We need to sort of come up with faithful reconstructions. Another sort of problem is that the data sets can be quite large, so there's a lot of computational complexity. That's where the Anvil comes in and Galaxy comes in, where there's a lot of tools that are necessary to make this efficient. You're not going to be assembling genomes by hand. We need highly optimized software working on big servers, big compute farms to be able to make this run efficiently. And then finally, once we get a possible solution, how do we know it's accurate? There's a lot of great tools to be able to do that assessment and then help you iterate to polish out errors, come up with a more complete and accurate representation. But in terms of the actual conceptual process, it was exactly what we heard for Charles Dickens. We're going to take our genomes here, instead of accidentally falling in the paper shredder, on purpose, we're going to be taking molecules of DNA and then shearing it into small segments. And that's for the very practical consideration that sequencers cannot actually read entire chromosomes end to end. They can only read off snippets, especially in the case of Illumina sequencing. You're only going to be reading off a few hundred nucleotides at a time. So we have to chop up our DNA molecules into the short snippets, 200 or 100 or a few hundred nucleotides at a time. Then we compare those fragments to each other using the Bruin graphs or other graph structures. That'll build this initial graph that we can pack down, and then we're going to try to detangle that graph using other types of information. So this could be, maybe you have a genetic map, maybe you have optical maps, maybe you have other sequencing technologies, long-read sequencing, mate pairs, high-C sequencing, to try to detangle that graph back into a complete reconstruction. So if you want to sequence a genome, there's a few ingredients that you just absolutely have to have. Number 1 on the list is you have to have sufficient read coverage. If you're sequencing a genome, like a human genome, that's about 3 billion nucleotides long, you should not just sequence 3 billion nucleotides. You need to oversample the genome quite substantially. And this is because that shearing process, that sequencing process, is a random process. And then if you just sequence 3 billion nucleotides, the best that you can do is get the reads next to each other without actually overlapping. The way we reconstruct genomes is by having overlapping fragments. So we need to oversample the genome enough so that we're guaranteed, sort of in a statistical sense, that those fragments are actually going to be able to overlap with each other that we can piece it back together. In round numbers, the amount of oversampling that you want to do is about 30-fold oversampling. There's some statistical analysis that we're going to talk about in a minute that sort of is the argument for why you need 30-fold oversampling. Another thing that you need is sufficient read length, and critically so that we can get through those complicated repeats. Right, in the limit, if our reads were only 2 nucleotides long, AA, AC, AG, AT, you know, we could build a graph, but it would be non-informative. Everything would be connected to everything else. It'd be a tangled mess. What we want to do is make sure our reads are long enough that we can get through these repeats as best as we possibly can. In this example, if we had longer reads, we would know those green nodes are connected to those orange nodes, or those blue nodes are connected to those pink nodes, and we can get through those red repetitive nodes in the middle. What read length do you need? Well, it depends. Microbes tend to have relatively small repeats, whereas mammalian genomes tend to have much larger repeats. In a mammalian genome, the most common repetitive elements are lines and sign elements, these transposable elements. They tend to be a few thousand nucleotides long. So in the here and now, if we can generate reads that are, say, 10,000 base pairs long, you can come up with a remarkably complete and contiguous reconstruction of the genome, as you'll hear about next week from Adam Philippi. And then the last thing that you need is high quality reads. If your reads have a lot of error from them, it's going to be difficult to figure out how they overlap, how they relate to each other. You're just not going to have a very successful reconstruction, and the reconstruction that you might have there could have a lot of errors in it. The good news is the software has gotten very sophisticated that we can overcome a lot of random sequencing errors just statistically in software. Although, you know, if the error rate is extremely high, this is just going to be kind of a hopeless endeavor. So in the interest of time, I'm not going to go through this in great detail, other than say Illumina Sequencing is basically a very, very fancy digital camera that can take pictures of DNA molecules that have been labeled with fluorescence dyes. So we can take our molecules, we prepare them, where we have strands of DNA, where we're going to synthesize complementary strands with fluorescence dyes of complementary nucleotides. We shine a laser on it, it'll glow different colors. We can then do image processing to decode those colors back into nucleotides ACGT. So Illumina Sequencing is a very fancy digital camera, followed by some image processing that can interpret those color dots to give us huge quantities of DNA sequencing. If you have a little time this afternoon, I encourage you to watch this YouTube film that Illumina generated. It's a little animation that walks through this process in great detail to really explain how it occurs. There's no magic to it, although it's a marvel of molecular biology that they've been able to scale up this technology that can sequence DNA so rapidly with such great accuracy for billions and billions, for trillions of nucleotides in a single sequencing run. One special part of Illumina sequencing is so you're going to be sequencing colored dots, which will tell you the nucleotide sequence. Now, we would love to just keep sequencing over and over and over again, but eventually the quality deteriorates and the molecules break down such that we can't actually sequence those colored dots forever. But there's a few molecular tricks that you can do to generate not exactly long reads, but give you more information about the genome. One is called pair-dense sequencing, where we're going to be reading off those colored dots, and then we flip the molecule upside down, and then we read off those colored dots from the other end. That'll give us a pair of reads, ACGT nucleotides, that both come from the same molecule, but they come from kind of opposite ends of it. This is pair-dense sequencing. One will be oriented on the forward strand, one will be oriented on the reverse strand. They are physically linked together, and that they originate from the same molecule. There's a sort of a turbocharged version of this protocol called mate pair sequencing, where again, you get a pair of reads, but the pair of reads comes from much greater distances apart from each other. And the protocol for that is we're going to take like a 10,000 base pair molecule, and we're going to actually turn it into a circle. So we're going to take, what is a linear molecule turned into a circle. And then what we're going to do is we're going to isolate the molecule right at that junction. So it'll be a few hundred nucleotides right at that junction. Now we're going to sequence from that junction sequence a pair of reads. So they're physically linked again, came from the same molecule. But with respect to that original molecule, they're 10,000 base pairs apart, although do this trick of molecular biology, they're going to be right next to each other. So that's called mate pair sequencing. It's a way to generate a pair of reads that can be separated up to about 10,000 base pairs apart. Although with respect to the original molecule, they point away from each other, although in the sequencing, they point towards each other. So there's, as you'll see during the exercise, you have to be very careful about your orientations. Paired-end reads point at each other, mate pair reads point away from each other. There's some special settings that you need to do for that. The data actually get written out to what's called a Fastq file. Again, the raw data is this image data of colored dots. Base calling software processes it, translates it into this text file of just ACGT. That's a Fastq file. This is how genomic data are represented. It's just a text file. You can open it in a text viewer, although they're enormous, where it's just read after read after read. In the case of paired-end sequencing or mate pair sequencing, you get two files. And then it's set up such that, you know, the first read of the file in both file describe both ends of the data, then the second read, then the third reads, then the fourth pair of reads, the fifth pair of reads, and so forth. So I just wanted to give you a little insight into the data files that we'll be working with. Now, I'm way over time, so I'm going to zip through this, but the key consideration is coverage. Again, it's a random process. These reads are going to be selected randomly from the genome. So the process is we want to generate enough data so that we sample every position in the genome, but we don't want to oversample too far because that costs money. It costs money. The analogy that I often use is imagine raindrops falling on a sidewalk. Each raindrop is just a drop of water. It'll fall, it'll land. Even though each raindrop is very small, eventually we can fill the whole sidewalk or we can fill the whole ocean, although we need a lot of rain to do so. And that rain, that DNA sequencing costs money, but it works out that we need about 30x coverage. This is an analysis of the Poisson distribution that tells us that we need about 30x coverage such that in a diploid species like human, we're going to cover both alleles, the maternal haplotype and the paternal haplotype with very high probability. It's a statistical argument. You know, we might get unlucky and parts of the genome will still be missing, but if you go to 30x coverage, statistically, you're guaranteed more than 99.999% of the genome will be covered to high quality so that we can actually reconstruct the whole genome. Now I'm running slow, so that was sort of the concepts. Let me jump ahead a few slides where we can talk about how these data are used in practice. And then you'll have these slides so you can go back and review the analysis. So we can collect about 30x coverage. We're going to build up these graphs. We're going to compact it down as much as we can. These compacted sequences will end for a variety of reasons. Maybe we get lucky, we've reached the end of the chromosome. Maybe there was a gap in coverage. Maybe there was errors in the data. Maybe there was heterozygosity, like in the case of human. The maternal and paternal alleles will be different. But crucially, these contigs, these reconstructions are going to end because of repeats where you just get confused about what's connected to what. And to overcome that, that's where the mate pair data, the optical map, the genetic maps are going to come in. So if we get this initial graph of A, B, C, D with repeats, if we know that A and B are a certain distance apart and B and C are a certain distance apart and C and D are a certain distance apart, hopefully we can come up with a unique solution to this graph so that we can actually lay out all the sequences across an entire chromosome end to end. That's how the human genome is assembled. That's how all genomes are assembled, where we get the actual sequence information, build our initial contigs, and then through scaffolding, lay them out end to end into chromosomes. So it's a hierarchical process. You have to have enough coverage. You have to have reads that are long enough and mate pairs and other information that we can tie it all together. We're going to start with those reads, building our graph, and then simplifying the graph, tying it together, and then hopefully getting it end to end. For short read sequencing, Illumina sequencing, my strong recommendation based on thousands of genome assemblies is to use a genome assembler called Spades. That's what we're going to be using in a minute inside of Galaxy. For long reads, come back next week. Adam Philippi is going to be talking about the latest and greatest of de novo assembly with long reads to hear about that. So let me pause there. I know I rushed through that a little bit, but hopefully the concepts are clear where we can collect sequencing data. We want about 30x coverage. We can then de novo assemble computationally contigs to then get scaffolded into chromosomes. Are there any questions about that?
Not yet. There was a question about K-mers, which I will address later. So.
Okay, great. Okay, so let me
Michael.
just, yeah, go ahead.
I'm sorry, I had a quick question. It was about the branch point will remain, and that's because of constantly overlapping. That was earlier when you had talked about that.
Yeah, let's kind of review here. So if we go back to the Dickens example, I think that's the easier one to sort of visualize.
Right. My question is, what we're talking about currently at this point, will that help to eliminate the point from remaining, or does it continue to read over and over and over until it gets a precise reading? Is that the point of the reading?
Yeah, great questions. So these branch points are really fundamental, right? If you're using a de Bruin graph assembler like Spades or Allpast or Soap de Novo or Velvet and yada, 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 there's many such assemblers. They take Illumina sequencing reads, which are about 100 nucleotides. They chop them up into K-mers, say 25 base pair sequences, and they build this graph. And this graph, for very fundamental reasons, will have these branch points, especially because of repeats. Like here, in this Dickens example, we knew that of times it was that repeat in the original genome. So we walk into that repeat, and then as you come out of the repeat, in terms of the sequences, it's ambiguous as to how you should come out of it. Now here you can kind of figure it out because we know something about English. But it's ambiguous if you should go in, it was the best of times, it was the worst of times, or should it go, it was the best of times, it was the age of foolishness or wisdom. You get these branch points, and you're just not sure how they can be resolved. So what assemblers do is they resolve it as much as they can up to those branch points. Those are the contigs that are output. And then you use other types of information like paired-end sequencing, Maypair sequencing, or optical maps or whatnot to tie those together, hopefully into complete chromosomes. But if the repeats are too long or there's still too much complexity, you won't actually get entire chromosomes reconstructed end to end. You'll get parts of it reconstructed. These are so-called draft genome assemblies where it's not complete genomes. It's sort of a patchwork of different parts of it that have been successfully resolved.
Thank you.
Great question. It's very, very fundamental to the process. Okay, so let's just, the last 2 minutes here before the break, let me just sort of set up the problem, and then Dave's going to walk you through how to answer it. So in the exercise today, we're going to be assembling a microbial genome de novo, where we've provided, Illumina-like sequencing data. The species that it comes from has a very special history to it. So it was identified in California in a place called Mono Lake. This is a real picture of Mono Lake. It's one of these lakes that is extremely, has high salt content. It's like, it's just like, table salt, but to the extreme. If you've ever been to the ocean, where it's kind of salty, imagine the most extremely salt environment that you can picture. Now, normally this is very toxic for microorganisms. They don't like to live in such a salty environment because it really, it just really sort of changes the chemistry, makes the ion transport very weird. This is normally very bad news to be living in an extremely salty environment. However, some microbes can live there. And this one microbe, I'm mispronounce this, Halomonas, can live there. And in fact, researchers a few years ago, 2010, went to this lake, identified it, extracted it, cultured it, and sequenced it. Now, this was a very big deal 10 or 11 years ago. And here's the reason why. In the journal Science, which is arguably one of the most prestigious journals in the world, they argue that this bacterium was so special that it could grow without any phosphorus. It could grow in the presence of arsenic. If you look at the periodic table, arsenic and phosphorus are right next to each other, and they have somewhat similar chemical composition. Although this was an incredibly amazing result, and that's because the backbone of DNA is made of phosphorus, or it has phosphorus components to it. So if this thing can grow and live without phosphorus, it would be the most foreign genome on the entire world. In that it does not have regular DNA, it uses arsenic instead. They try to prove this by growing this in culture. in a medium that they thought had no phosphorus there, and they would feed it arsenic instead. They could see the cells dividing, and they wrote it up as a science paper. So they wrote it up, although there was a lot of criticisms about it. In fact, other people tried to reproduce these results, and they could not. Basically, the scientific consensus to this day is this microorganism actually does have phosphorus in the DNA backbone. It's required, although it only needs very trace amounts in the culture medium that they use. But nevertheless, it remains kind of an interesting and exciting organism. So what I provided for you in the exercise that we're going to do in a minute is simulated sequencing data from a novel strain of this Halomonas genome. And your goal there is to sequence the genome, compare it to the reference genome, and look for novel genes that are going to be present there. The way we're going to find these novel genes is using a process called whole genome alignment. We're actually going to use an algorithm called NUMER, or NUCMER for the alignment. We're going to be comparing one genome to the other. In the simple case, this is easy. Some parts of the genomes will have an exact correspondence from one to the other. Some parts of the genome are going to be a little bit more complicated, where you could have inversions, you can have insertions, you can have deletions. But basically what we want to do is do this whole genome alignment using the MUMR suite. And what we're really interested in are parts of your new assembly that have no correspondence on the reference genome. That'll be, you can think of this as like this novel insertion in this novel species. And then what we're going to do is we're going to look for gene sequences inside of that novel insertion where we can sort of decode the codons from nucleotide sequences into amino acid sequences to go look at the sort of protein sequence that is associated with this novel insertion. If all is going well, hopefully there'll be a really interesting protein sequence to be discovered there. So normally, if you wanted to solve this exercise, there would be a big rigmarole where you have to install the tools and get them configured. But because we're using Galaxy inside of Anvil, I've taken care of that for you so that you can just immediately get started importing the reference genome, do the assembly, and then go look for the foreign gene and then hopefully translate it. So that's what we're going to do in just a few minutes. Let me pause here and see if there's any questions. We're running behind time, but we had built in a break. I'm hoping what we can do is just take a shorter break and still get started again at 12:20. I just want to make sure we have time to get through everything. Are there any questions? Otherwise, let's take a quick break, and Dave will take it over to go through the exercise.
Okay.
All right, thanks, everybody. We'll see you in about 5 minutes at 12:20 to actually do the exercise. Thanks, everybody.
Okay, team. It's been 5 minutes. It's a short break. First of all, somebody nod their head if you can hear me. Yeah, thank you, Mike. Okay, because I can only see you, Mike, so that's good. Let's see. Let's change that view. And now I can see more people. Okay, good. So I'm new to WebEx. Okay. Let's get started. Yeah, let's do that. So we're now going to do the tutorial and the slides. The slides are screen captures of what's in the tutorial. And so I'm going to be walking through the tutorial, which is right here. Okay. It looks like this. And the slides are there too, if you want to link back to those. But we're going to start right here. Explore Workspace and Launch Galaxy. We already did that, except now we're at the Launch Galaxy part. Okay. So let's go back into Anvil. And when we left this 40 minutes ago, we had just launched Galaxy, and now it has gone through a couple of phases, and now it's ready to run. So let's do that. So we're going to start Galaxy. And now we're actually going to see what it looks like. Let's zoom in a little. Okay, that's good. Again, ask questions as we go. I prefer that model. So we're here in Galaxy. This is a standard 3-panel design for Galaxy. Over here we have the tool panel, which is where all the tools we're going to run are, and there are hundreds of them here. There's a search box we're going to use extensively today. Over here is the history panel, and that's where our analysis shows up. So our data sets are there. Every time we run a step, we get more added to our history. And in the middle is where we configure our tools and run them, and where we look at our data. So that's the basic 3-panel design. Since we're running in Anvil, it has launched us, and I am already logged in. And furthermore, I am logged in as admin. I don't think we'll have time to get to this, but that allows us to do cool things. So, okay. So let's get started. First thing we need to do is import data sets. So if you recall from the start, there were we had one data table in our Terra workspace. So we're actually going to do that. We're going to get information from that. So you want to click on this one, the upload, that little guy right there. So we click on that, and it brings this up. This is a standard form, except it has choose remote files. And we're going to click on that, choose remote files. And since we're running in Anvil, it knows that we are, and it knows about the workspace that we launched from. And there's my workspace, Clemens VADSTI. Okay, so I'm going to click on that. It takes me in there and says, okay, you have other data, reference data, and tables. Well, I didn't have any of this. All I have is tables. So I'm going to click on that. My one table was called sample. So I'm going to click on that. And then something interesting happens. So you recall there was only one table. And this is actually that table. I could download it as it is. But there was a special column in that table which had URLs or links. And those links pointed to data sets that are inside Google Cloud. And Anvil recognizes those as special, and Galaxy in Anvil, also recognizes those as special. So what I can now do is I can import those 5 data sets into Galaxy because they are linked to from one of my data tables. So I'm going to select all 5, 4, 5, click on each of those. I'm going to say OK. And then I'm going to say start. And a couple of things are going to happen very quickly. First thing, we get all green. That's good. I'm going to click close. Okay. So this went to gray, first of all. Gray means that Galaxy has scheduled the upload. Okay. And then it went to peachy, which means it's running. So it was actively importing the data into Galaxy. And then it went to green, which says, hey, we got the data. Okay. If it had gone to red, that would mean there was a problem. There would also be a little bug icon here. So I don't know. Let's poke this a little. So I just clicked on this top line here, the name, and it gives me a preview of what it is. It says it's FASTA. That's a standard format for DNA, Halomonas, and it has one sequence. And this is our reference. This is what we think Halomonas actually looks like. We're going to do an assembly of these 4 things and then compare it with this. Okay. I can view this data more by poking it in the eye right there, and it will bring it up in the middle panel, and then I can see what it is. Now, I don't read DNA that often, so I have, that's not that useful, but it's there. So I'm going to minimize that again by clicking on it. Okay. Now, before we get started, there's a couple best practices I want to do in Galaxy. One is name my history. Galaxy is quite happy to have you have 15 histories called unnamed history, but it doesn't make you happy. So if we have to do this 3 or 4 times to get it right, and we come back an hour from now and we have 4 unnamed histories, we're not going to know which one worked. Okay? So always name your histories. That's the first thing, best practice. Second thing is I'm going to put some tags on these. They're hashtags, and those tags will percolate up through our analysis. And this will really help us with keeping track of where data came from. So to do that, I'm going to click on this, Edit Dataset Tags. So click on that. And then there's a form element here that we can't see. That's not a feature, that's a bug. Click on that. And this shows up, but we can't see it. So click on it, and then we can see it. Okay. And I'm going to say paired end. Okay. Paired end. I'm going to come up here. I'm going to do the same thing. Paired end. Hopefully I spelled it the same. Yes, I did. Come here. I'm going to do that. And I'm going to say mate pair, because this is the mate pair. Mate pair. Okay. And these tags will actually help us as we go. Oh, that's not what I wanted. And boom, mate pair. Come on, Dave. There we go. Mate pair. Okay, and then this guy I'm going to label reference. That's, sorry, that's not quite as important, but it will help. Reference. Okay. So now I'm going to go up here to my cog. So I've got all my data sets expanded, which is good, but it makes that history hard to process. Clicked on the cog. I'm going to say collapse expanded data sets, which brings me back to here. Okay. So the first thing we want to do in our process is run a tool, and it's called Spades. Mike talked about this. Spades. So I'm going to type it in up there. And it turns out there are a lot of tools related to Spades, including Spades itself right there. I'm going to click on that. Okay. And this brings up a tool form. This is in a standard format for Galaxy. All tools use the same framework to configure them. And there are a bunch of parameters here. For our case, we are quite happy to use the defaults, okay, except for k-mers. Now, William asked a question. How do we know? In Mike's example, it was 4 words, so we had 5-word chunks and we set our k to 4. How do we pick a good value? Well, our value that we're going to pick is 31, and there is some guidance here about what it needs to be. It needs to be odd, for instance. It needs to be less than our shortest reads. So the mate pairs are only 50 bases long, so it needs to be less than 50. And how do you pick k? Like, why is 31 better than 49 or better than 1? Well, Mike talked about Mike talked about how 2 or 3 would be really hard. 49 would also not take advantage of the assembly because you want to make the pieces small enough that you're likely to get overlaps between different reads. And if you make them the length of the whole read, you're unlikely to get like a 49 base overlap between a lot of reads. But 31, that's a good number. You are likely to get a lot of overlaps where 31 bases from one read actually overlaps with 31 bases from another read. Okay, so that's how 31 is picked. Yeah, so that's that. Okay, so I'm not changing anything else at the top. I come down here, 31. And now I say, okay, what files, what reads am I going to use to do this assembly? I have two libraries. I have this, the paired end, and this, the mate pair. And that's what I'm going to specify. I got paired end, that's the default. My orientation is that, okay? That's good for paired end, okay? And then I'm going to specify what my files are. I have forward reads by default, and I have never seen this violated. The .1 is the forward, the .2 is the reverse. So we're going to label that. We come down here, paired end is what I want. I want the .1. I come down here, paired end .2. So right here, just having this tag is helpful to remind me what's what. So I do that, and now I need a second library, this guy right here, which is the mate pair. So I'm going to say insert library. I click on that, and it's not paired end, it's mate pairs, okay? And it's the other way around. Remember how Mike talked about how it loops back? And then we actually take the two ends of that loop. So we want that. And then separate files, same thing. And we want the forward, which is the 1. And we want the reverse, which is the 2. Now, here we only have the 2 libraries. We could have lots of libraries. We also don't have PacBio or any of this stuff, okay? So we are ready. Yeah, okay? So we're going to execute. I'm going to click execute. Every tool has this at the bottom. Okay, now while that runs, let's come back to spades. So I'm going to click on run this job again. I'm not actually running it again. What it's doing is allowing me to run it again. So by clicking on that, it brings up the tool form with whatever I ran it with, okay? So how did I pick spades, okay? Well, I picked spades because Mike told me. Mike is my boss, okay? But what if Mike didn't tell me? How would I find spades, okay? There's a number of ways. There's 2 resources I want to talk about. One is Stack Overflow Bioinformatics. So that's an online Q&A forum, which is excellent. Stack Overflow Bioinformatics. Second is BioStar, another online forum for Q&A, okay? Now, what if I didn't get a clear vision like, okay, I've got this bacteria and I want to do assembly, what should I use? Spades is a great answer for that question, okay? But what if I don't come clear with that? Well, it turns out that there's actually hints in Galaxy itself. So here I'm going to say show sections. And I have a section in my tool panel called assembly. And so if I had no other guidance, I could go there and actually look through what's in assembly. Spades is one of the programs there that I could actually run. So I might discover it, okay? Another thing before we move on is how do I learn about spades, okay? Because it's a sophisticated tool. And how do I learn how to use it? If you scroll down to the bottom of the tool page here, there is some help, okay? There is also some help in the form about how things work. But at the very bottom of most of these, there's also a link, okay? Here it's a link to a paper. That paper probably has linked to documentation about spades. Maybe there's a forum about spades specifically. Maybe there's a mailing list. And these are great resources for figuring out, okay, what does it actually mean when I say this, okay? What does it actually mean when I say, yes, they're ion torrent? And you can find that out through these resources. Okay, so the second bug, and I think the last bug of the day, is note that I put tags on these, and I said they would percolate up, but we don't see them. And that's because of a bug. So I'm going to click refresh history. And now we see them. So we're going to click that a lot as we go. And what this tells us is that the spades output here was built from these data sets. Okay? And again, it tries to help us keep track of what's what. And as we get more and more data sets over here, these become even more important. There is one thing I want to look at here, which is the contig stats. So I'm actually going to poke that in the eye. So Spades did an assembly. It came up with contigs, which are high confidence parts of the genome that it's pretty sure actually look like this. And it came up with 4 of them. Now it's a bacteria, so there's only 1 chromosome. It wasn't successful at building the whole chromosome, but it was successful at building 4 contigs. So 4 high confidence places. It was also successful at building a scaffold, which in this case I think is just 1. So it actually did come up with 1 chromosome, but it has some low confidence stuff that connects it, okay? So we're still pretty confident, but not entirely. Today we're going to use the contigs, these 4 things. One of them is quite long, the others are about half that. Today we're going to use the contigs in our assembly, okay? So before we move on, and let's see, where are our contigs? Our contigs are here. If I poke that in the eye, that's the DNA in each of the 4 contigs. There's 4 sequences here. Yes, okay. So that's the actual DNA. What I want to do is add a tag to this that says, hey, this is contigs, pay attention, okay? And so I do that, and I say contigs, okay? And then on scaffolds, I want to add one here, and I actually want to avoid this because I don't want to use scaffolds by accident. So I'm going to put scaffolds here. And if I see scaffolds at any point, I know that's not what I want, that I've messed something up, okay? So let's see. Okay. If there are questions, somebody let me know, okay? Okay, good. So we've got our assembly, and it produced 4 contigs. Now we're going to use something called NUCMER, N-U-C-M-E-R, to visualize what we got. Okay. So align 2 or more sequences. And it says, okay, what's your reference sequence? And that is ref right here. That's the standard definition of Hellomonas right there. And then what's your query sequence? And by default, it says scaffolds. And it says that because it's the most recent data set in my history that this can use. Note that it doesn't list 1 through 4 or data set 6 and 7 or data set 10. That's because NUCMER can't use that type of data. It can only use FASTA, okay, which is what these 3 are. So I want to change this to contigs. I do that. And then everything else, I think I want the default. Scroll. It's a pretty big form. Yeah, I'm going to give it a title. Why not? Plot title. Ref versus our contigs. And then execute. So all I really changed was this and this. Okay, and I should be running on contigs. I'm going to execute. This one has a lot of help at the bottom. So if I click on that, do that. Yeah, it had a lot of help at the bottom, and it points off to a GitHub repository, which means probably documentation. And as Mike mentioned, this is part of the Mummer package, which is a large set of tools for doing this sort of thing. Okay, so once you get this, we're going to look at the plot, and it's going to give us a visual idea of how our assembly lines up with the reference. Okay, and again, there are tags, and that irritates me, so I'm going to refresh my history, and now I have my tags. And good. This says contigs, which means I didn't run it with scaffolds by accident, so I'm pretty happy. And now I want to look at the plot. And so I poke it in the eye, and it brings this up. I'm going to minimize this so I can see more of it. So I minimize my tool panel. And let's see, but I can't see the whole height. That's great. Okay. Along the bottom, so the x-axis here is the reference genome. And then what it's plotting here is how do my 4 contigs align with the reference genome? And it turns out my 3rd contig aligns with the front. My 2nd contig aligns with the next little bit. My 4th contig aligns with this bit. And then finally, my contig 1, the longest one, aligns with the tail end. And I can eyeball this and I can say, wow, that looks pretty good. There's not a huge gap between these and, between this and this. So that's promising. I also see here on contig 1, there's this dot. And that dot is actually what we're going to care about because that's the difference between the reference genome and ours. Okay, so we'll come back to that. So what's going on here. And to find that out, I'm going to run a tool called DNA Diff. Oh, it's already there. Sorry. Oh, didn't mean to do that. So when I typed in NUCMR, it was in that list because DNA, yeah, anyway, it was in that list. But I could have typed in DNA Diff and I would have got it. It's going to evaluate similarities and differences between two sequences. I'm going to click on that, okay, because that sounds like what I want to do. Same question, what's my reference? My reference is that. And then what's my query sequence? And again, it scaffolds by default. That's not what I want. I want contigs, okay? And then I really hate this question. Output only the general report file. Yes. Well, I want all the files. So to get all the files, I say no, which in my brain makes no sense. That's how the tool is defined. So please get me all the output files that this guy produces, because there's one in particular that I want. So click execute. It goes off and it produces a whole bunch of things. Now, I can see the future because I've done this before. I know that QDiff is the one I want, but there's also RDiff, there's SNPs. Maybe there were SNPs here. It turns out there's not. There's just one insertion. There's a whole bunch of different outputs here that I could look at, okay? But QDiff is the one I want to look at. So I look at that, poke it in the eye, and it comes up and it tells me there's something going on with node 1. So that was my long contig, and there's a gap. And in particular, it starts here and it ends here, and it's 333 bases long. So I've got an insertion of 333 bases in my contig 1, which was indicated by the plot. It's also found by DNA Diff. This is all encouraging. Okay, a couple things I want to do now. I want to use this information to extract out that DNA, those 333 bases. And there's a couple things I have to do first to do that, okay? So that's what we'll do now. Before we do that, there's a couple of cleanup things I want to do. I'm going to add a tag here, say difference, because now I'm doing stuff with differences, okay? So I fold that in. Something else I want to do, again, if I didn't know what was happening, I would look at all these, but right now they're just noise. So I'm going to delete them. Delete, 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 delete. And that'll keep my history cleaner and more tractable in my brain. Okay, and that one. So that's great. What happened if I got overzealous and I accidentally deleted this one too, which I need? Well, the data sets are still there. If I click on that, they're there and I can undelete them, okay? Or I can permanently remove them from disk, either one, okay? So if you accidentally delete something, don't worry, it's still there. Okay, so we got that. Now we need to get this into something. Yeah, we need to use this information to extract the DNA. We're going to use bed tools to do that. And to use bed tools, that's another suite like Mummer, which is a lot of tools. They operate on bed files. We need to convert this information into bed format. And we're going to convert it into a 3-column bed file. And a 3-column bed file, so bed is a standard format for genomic features. It's going to have the name, the frame of reference, which in our case is the contig, in the first column, the start position in the second column, and the end position in the third column. So we got to get rid of these columns here. That's the first thing we got to do, okay? Excuse me. And to do that, we're going to use the cut operator. And what cut does is it slices it slices tabular data. And this is what this is. Tabular, that means it's tab delimited. So there's tabs between all these. And we're going to say which of these columns we want to hang on to. And we want to hang on to the name. So QDiff, good, that's the one I want. And I click down here and I want column 1, that's the name. And I want column, let's come over here, I want column 3, which is the start, and column 4, which is the end. Okay, so I click here and I want column 3 and column 4. And what this is going to give me is just those 3 columns. I click on execute. It's queued. It's going to run, and then I will get back a 3-column data set. Okay. And I poke it in the eye to see it, and it looks like I got what I wanted. I got the name, I got the start, I got the stop. Okay. And so I do have a 3-column data set, and so I can run bed tools, except I can't. And this is because of the fundamental question in bioinformatics, and I am sorry to say this. And that question is, should we start counting at 0 or should we start counting at 1? And there are actually good reasons for this debate, but most of the time it's just tedious. And it turns out that DNA diff counts starting at 1, and bed format and bed tools, therefore, starts counting at 0. And so we have a problem here, which is that both of these are off by 1 in bed nomenclature. So we need to move them from 1 relative to 0 relative. This needs to become a 23, and this needs to become a 55. Okay. I love this example, and Mike created this example because it highlights both how cool it is to do analysis and get cool results, and how you can string together all these tools. It also highlights how tedious parts of bioinformatics are. And, one-off errors are about as tedious as it gets. Okay. So we're going to fix this. The tutorial has 2 ways to fix it. I'm going to use awk because it's quicker. And let's talk about awk. So I type in awk. It's under text manipulation, and that's what I'm doing. I'm changing these guys by 1. Text reformatting with awk. I'm going to take a cough drop because I'm going to start coughing. So I'm going to talk with a marble for a bit, and I apologize. Okay, awk is this incredibly powerful and flexible program, and it allows you to do all sorts of really cool manipulations on tabular data, and that's what we have. It turns out that our manipulations are pretty simple. And if I scroll down here, it tells me, well, cool, there's stuff I can do. So for instance, print 145 will print only the 1st, 4th, and 5th fields. Print 4, 5, minus 4 will print column 4, and then it'll print 5 minus 4, the columns. Well, this gives me an idea of what I can do. And this is also in the tutorial, so you can just cut and paste it. But this is sort of what I want to do. And so I'm just going to put it there. And what this says right now, anyway, is print column 1, column 4, and column 5. Well, I don't have column 4 and column 5. I have column 2 and column 3. As of right now, it's going to produce the input. Exactly. It was not quite what I want. So I say -1, -1. Okay. And again, you can cut and paste that from the tutorial. And it says, take the input, print it out, except take 1 off each of these. I'm going to click execute and then come back. Okay, so go there. Now, awk is one of the options we show here. The other option described in the tutorial is table compute. Table compute does not force you to learn this syntax, okay? It's a very flexible and powerful tool as well, but you don't have to learn this syntax. You do have to learn the tool. Now, here you have a fundamental choice, and Galaxy tends to support both of these in lots of different situations. You can use a tool like awk, which is defined in Galaxy, but you have to learn a special syntax, okay? Now, once you learn it, you figure out, okay, it's not actually that hard to do basic stuff. I have learned awk 15 or 400 times in my career. So I don't remember awk, how it works, but every time I need it, it's like, okay, I can figure this out. Same thing with table compute. It's like it takes a bit to get the hang of it the first time, but once you get the hang of it, it's easy to relearn. So which of these should you use? It's entirely up to you. If you're comfortable learning special syntax like this, use awk because it's faster to type that in. If you're not, then use table compute because you will get the same results. And you can also do very cool things with table compute. You could do this with table compute. Okay. So here we go. We got this. Let's take a look at, so this is the output of awk. It's got 39923 and 40255. I'm going to open this guy. And we started with 24. We're now at 23. We started at 56. We're now at 55. So it looks like it worked. Okay. I could run table compute now, and I should get exactly the same results here. In the interest of time, I'm not going to do that. Okay. So now what we have here is actually 3-column bed, and it's 0 relative. So it actually is a bed. So I'm going to go, cool, I want to run bed tools. And the particular tool I want to run is called get fast. Get fast is enough. Get fast a bed right there. Okay. Use intervals. That's what this is. Those 3 things form an interval to extract sequences from a FASTA file. And what I'm going to do is extract sequences from my context file. So cool. I click on that and I go here and I'm excited. And it says, Dave, you don't have a bed file. And I go, well, crap, I do have a bed file right here. Well, yeah, okay, it is a bed file, but we're not telling Galaxy that yet. We need it to now tell Galaxy this is bed. And the way we do that is we change it from tabular, which it is, to say, well, it's tabular, but it's also bed. It's a more specific type. So click on Edit Attributes, and then go to Data Types, not Convert, because this is exactly what we want. We don't want to rearrange things or change things. We go to Data Types, and we want bed, B-E-D. Okay. And then I click Change Data Type. Okay. And now, when I launch this tool, it will actually recognize this. Okay, so Galaxy goes through and it looks at it and says, hey, you're right, it is bed. Okay, cool. And now I go to Get Fast A, and now it shows up. Okay, so I didn't add a data set, all I did was change the data types, but all tools in Galaxy know about data types. So we're good there. Choose the source of my history. What's my FASTA? Again, I don't want scaffolds. I want to get these 333 bases from my contigs. Click on that. And then I think I'm done. Yes, I am. Okay, so that contigs, good. That's the only thing I changed. And I click Execute. Okay. So it's going to run, and should run pretty quickly because there's only 333 bases. So now let's poke that in the eye. Okay. So I'm really close to the problem as Mike defined it, which is we want to know what amino acids are produced by this insertion. So here's the 333 bases of DNA. I hope it's 333. I could run another tool that says, count how long this is, just to confirm I got 333. But I got my 333 bases, and if I were a good biologist, I would know what GGC, resulted in as an amino acid, and AGG, and all this, and I could just look at this and go, aha, it's whatever. I'm not a good biologist. I'm a computer person, so I can't do that. So the next thing I need to do is run a tool that says, hey, here's some DNA. Tell me what the amino acids it will produce are. And so to do that, I'm going to use an oldie but a goodie called Transseq. Transseq is part of the Emboss package, which has been around since before I was in bioinformatics. Okay, so it's really a lot of very useful tools here. I want to try Transseq. If that doesn't work, I'll go to BackTransseq, which reads the opposite side of the DNA and does that. Okay, but let's start with Transseq. I click on that, and I'm here. Okay, and is this what I want? Bed tools, get fast. Yeah, that's what I want. Okay, good. And then I don't want to change the frame. I want to start exactly at position 1 or position 0. Take your pick. And I come down here and there's nothing else I want to change. Oh, there is. It doesn't matter, but we're going to do it right, okay? Because we're biologists. And so I don't want to use the standard table. I want to use the bacterial, which is right there. Okay, we get the same result, by the way, but sometimes we wouldn't. So I want to use the bacterial translate DNA to amino acids table. Okay, and I want to do the whole thing. And now I'm going to click execute. Okay. How am I doing for time? I'm pretty good for time. Holy cow. Okay. Yeah, this is exciting for me because that means I get to talk about cool optional stuff. Okay, that's going to run pretty quickly. And now the big reveal. Greetings, VADSTI, from friends at Anvil and Galaxy. Keep searching for those little green men. Okay. So this is an excellent protein. And I can tell you that this bacteria must have been raised, excuse me, under selection pressure for VADSTI participants to say this is a good result, because otherwise we wouldn't get this, okay? A couple things to note here. The stars are actually stop codons, so what we're getting here is a lot of little proteins, okay? And Mike has replaced some things here because there's no O in the translation table. So there's a Q and there's X's and yeah, or K there, sorry. So we now have our result. We started with this sequence from this experiment we ran, and we wanted to know what's the difference. And we walked through it. We ran, I think, 7 tools to do that. We had 5 inputs, and we got to this point, okay? Now, before we do any of the optional stuff, because I have time, before we do any of the optional stuff, what do we want to do with this? So we want to save our data so we have some results. And I'm going to argue, maybe I want to save this, maybe I don't. You know, maybe I should just save the DNA because I can always get this from the DNA. But I'm going to save this, the DNA, and the contigs are the 3 things I'm going to save. So I'm going to click on that, download. Comes up, downloads it down here. Okay, I'm going to save this one. Click on this, download. Shows up down here, good. It's now on my laptop. And then I scroll down here until I get to the very first contigs tag, which is not there. Nuckmer, spades. Where are you? Oh, yeah, refresh. Oh, I hate that bug. Okay, right there. That's it. So I want to download that too. So I expand it and I click on that little diskette, download. And now I have my contigs and my DNA and my proteins, okay? And now I could shut this down, okay? And that's what we'll do at the end, but I think I have 15 more minutes to talk, so I'll do that. I could shut this down now, and we will do that, but there's 2 optional things I want to walk you through before we do that, because I have time. That is so exciting. Excuse me, are there any questions? No. Good, okay. If you don't wait long enough, then people don't have time to ask questions, and you just get to go on and cover what you want to talk about. So let's do that. Okay. So I'm going to collapse these again, collapse expanded data sets. We've got, now I deleted 6 or 7 of them, but we've got like 18 data sets here. And the tags are actually helpful for knowing how things relate to each other, because you can follow them through, but it's easy to lose the big picture. And if I didn't have a recipe to begin with for how to do this, I would try all sorts of things, and maybe I would remember to delete them, and maybe I wouldn't, you know, until I arrived at my recipe that I actually want to use. So in reality, this is going to be a lot messier than what it was today, because today, thanks to Mike, we knew exactly what to do. But in reality, you're going to try this, you're going to try that, it's not going to work. I'm going to show you a trick for getting a better picture of what we actually did. Okay. So in Galaxy, there's something called workflows, and I'm going to click on that. And I could show structure, but that doesn't actually show what I want to show here. What I'm going to do is extract workflow. So I'm going to click on that. Okay. And it gives me a whole bunch of stuff here, which I'm going to ignore for right now. And I'm going to say that's a great name. Okay. Just create it. What a workflow is a repeatable recipe for doing this analysis again. So I have now created a workflow, and you know, if I have another sample, excuse me, of a different bacteria that was under different selection pressure, I could upload that. I could create a new history and upload those 5, and then rerun this workflow against that. And I wouldn't have to type in spades and Nuckmer and all that. I could just say, here's my 5 inputs, run my workflow. I'm not doing that today, but what I am doing is using the workflow editor to show the structure of what I did. Okay. So let's see, and I'm going to shrink my zoom some so I can actually see everything. And I'm going to minimize this. So what I clicked on there was edit my workflow. Okay, and up this comes, and I will admit that this initial rendering does not offer us a lot more insight than we had from that stack in the history. But what we can do is rearrange things here. And I'm going to do one more zoom out. And so what I've got over here are my 5 inputs, the ref, the paired end, I'm going to do that. Okay, I'm going to do that. And then I'm going to drag things around until things start to make more sense. So I'm going to drag my spades down there. So those 4 things go into spades. That's good. The reference did not, apparently. Okay. Let's drag that down a little more. I'm going to put my reference up there. And then it looks like, yeah, okay. Drag that over. Okay. Now, with just a little bit of playing, I've been able to see what this workflow actually does, what I did in my history, which again, it's easy to lose track of while we're in the heat of it. I had 4 inputs here, which got fed into spades, and they were as 2 libraries. Good. Spades produced 1, 2, 3, 4, 5 outputs, of which looks like I used 1. Now I can tell I only use contigs here. I never use scaffolds because there's nothing coming out of here. Okay. And then what did I do? I used NUCMR with my reference and with the output here. And all I did was look at it. So I didn't use the output from NUCMR again. Okay, that's interesting. I only used it to visualize things. I could have used the output from this guy, but I didn't. All I did was look at this. I then ran DNA diff with the reference and the contigs, good, and it produced all these outputs, of which I ignored all of them except this last one. And then I did 1, 2, 3, 4 steps to get the DNA out. Okay, so this structure is not at all clear in that history view. Now we're working on that, and we expect a release later this year to actually address how the history is presented. But in the meantime, this is really a great way to figure out what's going on here. Okay, so this is workflows and the history view, or yeah, workflows and the edit view. You can also create workflows from scratch in this view. And so, for instance, if I had if I wanted the output of this advanced cut to go into a filter or a sort, let's say a sort, I click on sort, I do that, I connect this. Okay. And now if I save this workflow, it's going to have this extra step in it. And maybe I do stuff from there. If you have a clear picture of what you want to do to begin with, you can create a workflow from scratch in here. I never have a clear picture of what I'm going to do when I start. So I always create my workflows from my history. Okay. I don't want that sort, so I get rid of it. Okay, so let's get rid of it. Now I'm going to save it. So I now have a workflow here. I come here and I can see this guy and I can view it again in edit and it will be there. And I can now run it again the next time I have 5 data sets. Okay, it's the same one. Okay, so reusable and a great way to view stuff. I got 12 minutes left. What else do I want to do? Let's go back to analyze data. Any questions? Okay, good.
Dave, there's a question in the chat about sort of exporting workflows, sharing workflows.
Yes, thank you. So if I go here to my workflows tab, it is possible. So download right there. It is possible to export your workflows. And right there, it just exported and it's in called, well, yeah, it's in what's called a GA format. And that I believe is XML. And I can now go to a different Galaxy server and import that. Okay. So it is possible once you create a workflow to also export that. What I can't yet do in Anvil anyway is export the entire history, all 25 data sets. Okay. That will be here later this year. Okay. On most Galaxy servers, you can now export your whole history, but not on Anvil yet. Okay, that'll be in the next release. Thank you for that question. Yeah, so it is possible to save this workflow And in Anvil, that's particularly helpful because you don't want to leave this Galaxy server up and running for a month while you don't use it. Okay. You want to come back and relaunch it a month from now. And unless you paused it, you're going to start from scratch, and then you can just upload this workflow. That's a great question. Thank you. Other questions? Okay, I got 10 minutes. Let's see.
Yeah, a quick question, please.
Yes.
Yeah. So your workflow, is it like an XML file, or is it a text-based file? Is it something that you can create outside of? Let's say I have a workflow that I've gone through the clicks you've gone through, and then I've gotten it. Well, this is nice. Can I then export it and then have that as a base workflow from which I can create another workflow outside of the system and then import it back and let it run? All in the interest of saving time and also avoiding the clicks. Having a text file with everything visible allows me to actually see what's happening instead of click, 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 and not seeing what it is.
So William, I'm going to try to answer your question. It is a text file, okay, and it is semi-understandable by a human, okay. I think it's XML. I'm not positive of that because I never look at them, okay. And high-end users actually do manipulate the text file itself, okay. But everyone I know, well, not everyone I know, but every biologist I know who's not a computer techie hardcore person, they manipulate the workflows in the editor that I just showed, okay. But you can edit it while it's been downloaded as a text file. And there are programs, so people write programs that do that, or they just edit the workflows. But in general, people use the editor, and that editor is available on all Galaxy instances. Okay. Cool. Thank you, William. And did I answer your Kmer question? Okay.
Please, yes, please. So we have something like that in machine learning where you try to run K from 1 to n and take the elbow, what they call the goal.
Yes.
And we learned that in past lectures. So this really sets in. So in this case here, the K is, it's a matter of you having an idea of a good idea of the data. You don't do much trial and error because it's going to cost you. It's going to cost a lot of money if you're going to run all trials of K to determine the best K, right?
Mm-hmm. True. In this case, it's not a lot of money because it's a small genome. But if you're doing like human, 3 billion bases, yeah, it's a lot. But here, we could have run multiple, and then we could compare the contigs to see, which one produced the least contigs, or there's a bunch of metrics you can run. We didn't do that today. I took Mike at his word, which, again, he's my boss, so always do that, okay?
No, but you're absolutely right. If we're going to do this for novel species, you often would try a few values. I mean, the extremes are, that, 2 base pair Kmers is too small. For 50 base pair reads, 49 would be too long for the reasons Dave explained. But there's sort of this, range of possibilities. In practice, they're going to be very similar. Maybe things will connect a little bit. It's all about getting through those repeats. So if K is a little bit longer, you'll be a little bit more successful getting through repeats. But in practice, there can be error. There can be other kind of weird stuff. So you don't want it to be too long. There could be, the reads themselves are random where they are positioned. So in practice, we'd often try a few values and kind of pick the best ones. Spades does have a mode where it can do that for you, where you can let it run a few values and it'll try to combine them in clever ways. But for this simple example, we're trying to get the concepts. We're trying to work through the steps of it. I should say, everything we did today was real. We really do assemble and publish genomes with spades. We really do align them with Mummer to look for differences. We really do extract the differences. We really do, translate the nucleotides back into proteins. The only kind of silly thing was having the secret message in English. Obviously, you wouldn't normally see that, but it just made it hopefully crystal clear that you knew that you had the right answer. Notice if you had been off by 1, you would have had a frame shift error and you would have gotten this scrambled up amino acid sequence. But here it was just kind of for fun to have a little secret message there.
So the first time I ran it, I got scrambled because I added one instead of subtracted one. So, okay. Thank you, William. Let's see. So I have one more thing I want to highlight before we shut down, and that is a cool thing about Galaxy on Anvil. So if I had run, so I only ran awk, but I could have run awk and I could have run table compute. And maybe I wanted to check if the output was different or the same between those to see if I'm doing it right. Maybe I want to check that my output between the cut and the awk is actually different. Now here I had 1-line file, so I could visually do that, okay? But if I have a 1,000-line file and I'm only changing very select rows, rows with particular conditions, I want to actually be able to check, did I change my file between one and the next? Now we did one tool that compared the assemblies to see what's different. There are lots of tools in Galaxy that compare different formats, but I want one that just looks at tabular data files and says, is there a difference? And it's not installed on this server by default. So what I want to do is go to the Galaxy toolshed and install it. So let's do that. Again, I'm logged in as admin, as you will be, because this is Anvil. So I click on the admin interface. And now I can do major harm to the server, although not to the world. Don't worry. You can't make the smoke come out of the internet. You could conceivably make smoke come out of Galaxy if you do the wrong things here, but what we're going to do is pretty straightforward. I'm going to go down here to tool management and say, okay, I need to look for a difference tool. So I click on install and uninstall, and then I search for Diff, because I know that's a Unix tool, just like cut is. And I got cuff diff. Okay, that's a specific cuff norm. I have all these things which are specific to particular formats. I just want the Unix diff. Can I find that? And if I scroll down, eventually I will find it, I hope. Boom, boom, we have a lot of diffs. Wow.
I think I saw it. You went past it.
Thank you, Mike, because I thought I would see it by now. Okay, well, F diff, D-I-F-F, that'll help me. Right there. Okay, not as hard as I thought. Okay. Written by Bjorn Gruning. Good, good, good. Bjorn is a superstar. So I look at that and I go, that's cool. And I click on that and it tells me, Dave, this is what you want. And I go, great. Looks like there are 1, 2, 3, 4 versions of this. I want the latest because I want to be hip. And I just click install there. And it comes up and says, Dave, where do you want it? And I think I want it under text manipulation. Filter and sort, maybe. Okay, it's not going to manipulate it. It's not, but I don't know. I'm going to put it under text manipulation, even though it doesn't actually manipulate anything. And I'm going to say, okay. And what's going to happen now is it's going to install Diff on my Galaxy instance on Anvil. It goes through cloning, which says, okay, I'm working on it. And then it will come up and say, hey, it's there. And you can uninstall it if you want. Okay, so now it says, Dave, it's there. Okay, well, I don't see it, but it says it's there. If I change my mind, I can come back here and uninstall it. So I'm going to go back to my Analyze Data view here. And I said, put it under text manipulation, and there it is. Okay, now, if we go back to here, 6,320 tools in the toolshed are compatible with this release of Galaxy. There's a lot of tools here. Chances are, if you want to use a popular tool and it's not here, chances are it's here. It's in the toolshed. So on Anvil, this is a really powerful thing. I'm now going to go back and I'm just going to run it to demonstrate that it actually works. And I'll show you one thing I didn't do. Okay. Transseq on data 24, that's not what I want. What I want to do is compare my text reformatting on data 22 with my cut to see if it actually changed it. Okay. And I say, no, I don't really want the HTML. I just want to see the text. And I do side by side. That's great. I do execute. And it now runs this tool that I just installed. I'm going to poke it in the eye as soon as it's done. Come on. Okay. And lo and behold, what does it say? It says, I got a diff. So I ran the diff against these two. Okay. Node 1 length, such and such. Node 1 length. Okay, so this was the first one. The pluses, this is the second one. It changed from 3 to 4, 5 to 6. Okay. So very simple tool. That's all it does. I could have said, please create an HTML report, which would have made this nice and spiffy. I didn't do that. But you can install tools on Galaxy by doing the admin and then go to the tool shed. Okay. So that, I think, is my last thing before I shut this down. Have we got any questions before we show you how to shut this down? Because once we shut it down, I can't show you anything.
Yes, I just posted Galaxy is mainly cloud. Oh, cloud computing. Okay. Yeah, I posted since you had Diff and you had other Unix-like utility tools available. I was wondering what OS is provided and how do you get to the prompt? Like on Codio, I can see the terminal. I have a terminal prompt. In Anvil, do I have access to the terminal prompt?
You do have access to the terminal. I have never used it, but it's not through the Galaxy interface. It's back through the Anvil interface. And I can show you that.
Okay.
Although I'm not going to go there. Where is it? There's a terminal right there. Okay.
Oh, okay. Okay.
Open terminal. And now I get really scared because, okay, I am a computer person.
Okay. Right.
Yeah, that's not what I want. Okay. And it already went kittywampus on me. So I'm not gonna touch it.
Okay. No problem.
But yeah, Anvil does allow you to get to the terminal.
Okay.
Okay.
Cool.
And
so now I can do things like FTP but basically when you say you bring the file over, let's say the IML or XML file that you exported, how do you bring it over? I mean, how do you bring it over? I know it's probably going to be around the export, but is it very obvious?
Yes. So let's see, is there FTP here? Okay, well, you can, I don't know if this server is FTP configured or not, but that is an admin configuration where you can allow your users to use FTP to transfer large data files. Okay. Again, I don't know if it's configured on this one or not, but if you do have it configured, users can use an FTP client to transfer it. And then when you do this, it will show up here. And the fact that it's not showing up here means it probably does not have FTP configured by default. Okay.
But
you
then
can change that.
You can do pick and drop. So I think, that answers my question. So you can actually do a pick and drop.
Yes, that you can do too. Yeah. Choose local files. You can paste and fetch. Paste and fetch, you can put a URL in. Okay. Or if it's very short data, like our one-line files over here, you can cut and paste the whole thing in. Yeah. Okay. But today we did that, and you can also
local files.
Yeah, you can do this.
I have not personally attempted to do the FTP thing, but if you can put the FTP file in, if you can retrieve it via the terminal in Anvil, if you put it in the same folders as Anvil retrieves, Galaxy retrieves file from Anvil, you should be able to go through a roundabout method.
Okay, thanks a lot. Thanks. Yeah.
Yeah, thank you, Alex. That was Alex Ostrovsky, who also works for Hopkins, also works for Mike, part of the team. So
thanks.
Yeah. Other questions before we close this guy down? Okay. Let's do it. So we're going to go back to Anvil. We've downloaded everything we care about. And we're back at Anvil.
Yeah, and Dave, just a quick question. So I remember you got a little frustrated when a view wasn't showing. Now, if you have access to these Unix-based tools, can you run these things in a batch? Can you set up like a batch job, which then runs and then renders your view with a single click?
You can.
Okay.
Yeah.
Okay.
Yeah. So we have an API and we have a Python library called BioBlend. And BioBlend will allow you to interface with Galaxy and do almost everything you can do from the graphical user interface, from the web interface. Okay. Things like interactive visualizations, which we did not touch at all today, you can't do because they're interactive. But you can fire off workflows, you can upload data, You can check the status of your workflows. You can do all that programmatically as well. So if you're a bioinformatician and graphical user interfaces just frustrate you, but you want all the pluses of Galaxy, the fact that it tracks everything, the fact that it keeps track of your parameters that were run, all the reproducibility features, all the authentication features, you can use the API. And that's what we recommend if you're a bioinformatician. Once you get your workflows set up, don't log in every Monday morning to launch the latest data sets. Just set up a cron job and it will check a directory. And when there's files in that directory, it'll load them, run the workflow, download the results. Okay. Now for Anvil, that's complicated because you pay for it in the intervening week. Okay. But you could easily set up an instance where you launch it through Anvil and then you fire off your, you fire off your scripts. Okay. Good. Thank you. Okay. Okay, so we're going to shut this guy down. And I'm going to go over here, click on Galaxy Interactive Environment. I clicked on that. And it actually gives me two choices here. I can pause it. And if I'm willing to pay, I think 9 cents an hour, that plus that, I think, then I could pause it. And I could come back a week from now and I will have paid 9 times 168, 0.09 times 168, okay? Or I can just delete it because I have downloaded my workflow. I have downloaded my data sets. I can get to the point where I was pretty quickly now. I'm going to delete it. And so I'm going to click on that. If I don't delete it and I just let it run, we're talking $12 a day, which is not much until you add it up over a month. And then you burn $400 that you got nothing for. So don't let it run unless you're using it. Pausing it is completely valid, 9 cents an hour, but we're going to delete it. So we click there on delete and it says, okay, Dave, if you do this, you're going to lose everything. Are you sure? It's like, yeah, I'm sure. Okay. And I click on delete and down it goes. And now when I come back, I can relaunch Galaxy if I want to. Go there and it will be a fresh Galaxy install. Okay. And we have 8 minutes to spare for questions and answers. So, Mike, have you got anything to add? Or sorry?
Yeah, Dave. So
Yes.
so the pausing and deleting, I see the need for that, but I'm really scratching my head. So when you fire up a Galaxy instance, it is an active job running. So even though you're not using it, it's still running. Now, when you pause it, why should you pay? Because now you're not using up any resources. You just, or is it that memory usage? I mean, what is the rationale behind having to delete it?
Yeah, it's the storage cost. Yeah, so
okay.
it's the storage cost, for the image of the instance plus your data.
Right.
Okay.
Yeah.
That makes sense. One thing I really meant to mention, was the credits program, which got mentioned yesterday, but I really want to push this. Okay. Thank you, William. Yeah, so it costs money. Okay. Now it's a great value. So 50 cents an hour for what we just did, that's a great value, but it does cost money. And if you're doing stuff in anger, not just toy problems, but you're talking about gigabytes and petabytes, and you're doing things that take a lot, which is one reason to use Anvil. It's going to cost real money. And so I really recommend you look at the Anvil Cloud Credits program. Okay, and you can apply for grants to support your research. Okay, so really think about this if you're looking at Anvil for hardcore research.
Yeah, Dave, I'm glad you brought this up. So this is a new program that we've launched, this AC2. The applications are due in about a week, but they're super short. It's just like a page or two, just giving kind of a broad overview of what you're intending to do. And then we intend to award up to $10,000 worth of credits. As Dave was saying, that's really critical if you're looking at large data sets. For a lot of human genome analysis, you have short read data, you want to call variants. It costs about, in round numbers, it's about $5 a sample. So that'll get you through a few hundred genomes worth of analysis for $10,000 worth of credits. If you want to do, develop new tools, that would also be a great application. If there's also sort of a training track, if you want to, teach classrooms with lots of people, kind of like we're doing today, where you have classrooms with lots of people where, you want to make sure that they can just come to the platform with no cost to them. That's another great use of this AC2 program. We'd be very, very interested for your applications. So that's in about a week. If you have questions, shoot me a note, or any of us a note. We'd be happy to work with you. And I think what's likely to happen is this is sort of our pilot for this AC2. I do anticipate in the coming months, there'll be other calls for proposals. I don't know if we're going to do it, once a year, a few times a year, but I do think this is going to be a regular event to offer these kind of credit grants very rapidly throughout the year.
Good. Other questions? Okay. Should we declare victory and end 4 minutes early?
I think we should.
Yeah, perhaps. Any questions for the Anvil team?
Well, thank you very much again for inviting us. Again, the secret message was kind of playful, but everything we did today was real. We really did assemble a genome using state-of-the-art tools. We really compared it to the reference exactly how we would. We look for those novel insertions. We do this all the time where it's, a novel virus insertion, a novel transposable element. We want to go and investigate that sequence. All of that was very real. These are exactly the tools that we would use to do this and publish on it. And then obviously doing it inside of Galaxy makes it all the more powerful for all the reasons that we heard about today. If you're interested to learn more, what I would also recommend is there's the Galaxy Community Conference coming up in just a few weeks now. That'll be a great opportunity where there's extensive training, there's research presentations. Again, as we heard, the most important part of Galaxy is the community, and we want to welcome you into this community. We'll help you, we'll support you, and then what I expect will happen is, once you get used to it, you become the trainer, and you can tell your friends, you can tell other people. It's a great environment to do lots of real sophisticated analysis and research. So thank you very much. We're happy to take any questions offline in the future, and I do really encourage you to apply to the GCC coming up in a few weeks.
Okay, thank you.
Thank you, everybody.
So, audience, as we do all the time, so put your cameras on and let's show our appreciation to the Anvil team, the presenters, and those who understand work tirelessly behind the scenes. So let's show our appreciation to them. Thank you so much. So this has been a very engaging, very informative presentation, and then we thank you so much. And as Mike is saying, you want to take advantage of all the resources, all the opportunities out there, so that you can become expert in this, and then you host the next training.
That's right.
Thank you so much, Anvil team, and thank you, participant, and then. Have a great weekend, and then we'll see you all again next week for the final presentation of the training season. Make sure also to complete the evaluation. That is very valuable to us. Your feedback is very important here. So make sure that you do that if you haven't done that already. Otherwise, thank you, and then we'll see you all next week.
Thank you.
Thank you. Thank you.
Bye now.
Thanks a lot.
Thank you.